Hi, my name is Nathan, and this is my presentation. I'll walk you through a 3D model I created for the COL1A1 protein, also known as the collagen type 1 alpha 1. Here's my model. These are the collagen fibrils, the white pieces of paper. This protein plays a critical structure in the human body, and mutations in this gene can lead to severe connective tissue disorders. One of those is Capi's disease, a condition affecting infants which causes painful bone swelling, inflammation, and overgrowth. The COL1A1 is not just important, it's essential for life as we know it, especially for the strength and integrity of bones, skin, and tendons. So I'm twisting this into a triple helix formation. So this is what it looks like. The COL1A1 is primarily found in extracellular matrix, this network of molecules outside the cell that holds the tissues together. It's produced by several types of connective tissues, including fibroblasts, which create collagen, skin, tendons, and ligaments; osteoblasts, the bone-forming cells that lay down the matrix of the bone; chondrocytes, which create cartilage, particularly on bone cartilage junctions; and odontoblasts, which help form dentin in our teeth. And the COL1A1 gene encodes for one of the genes in type 1 collagen, the most abundant collagen type in the body. It combines with the COL1A2 to form heterotrimer. Two COL1A1 chains and one COL1A2 chain makes up the full collagen molecule. This is an example of a quaternary structure. Now we're going to move on to the structure of this. And normally the COL1A1 model is built from 3 white paper strips twisted into a triple helix. This represents the coordinate structure of type 1 collagen. Each individual paper strip symbolizes a single polypeptide chain made of the sequences of amino acids, which is the primary structure. As these chains coil into the right-handed helix, you'll see the secondary structure forming. And the triple helix is the tertiary structure of the coordinate folding, which gives the collagen this incredible tensile strength, even stronger than steel per weight. I marked up the multiple arginine, or R836, residues along each chain with green colored dots. Arginine is a large partially charged hydrophilic amino acid that plays a huge role in ionic bonding and hydrogen bonding with negative charged molecules with glutamate or the phosphate groups of the extracellular matrix components. And this allows it to interact with fibronectin, integrins, and protoglycans, and even calcium hydroxyapatite for bone mineralization. Next, we're going to talk about the mutation for this. So here's the mutated version. As you can see, here's arginine, but it's replaced with cysteine, the C836, a mutation known as the R836C, linked to Capi's disease. In my mutated model, I removed the R836 dots and replaced them with the C836 labels using a different color. I loosened at the twist of the helix, so here's the loosened version of the helix. And this creates a less stable fibril to reflect misfolding. And here's the key. Cysteine contains a thiol group, unlike arginine. When the two cysteine residues are close together, they can form a disulfide bond. And a disulfide bond is basically formed when these two, the paper clip, forms an abnormal bond between. And then these bonds are not supposed to form here. They lock the protein in the wrong shape, which disrupts the collagen ability to assemble properly in fibrils and anchor the extracellular matrix. And basically this happens when we prevent proper extracellular matrix assembly, and it weakens the bones and tissues and triggers inflammation. And this can alter cell signaling because extracellular matrix also acts as a mechanotransduction platform, relaying external forces into cellular responses. So now we're going to talk about the structure. So for the primary structure, for the amino acid sequence in this case, the critical difference between arginine and cysteine at the position of 836. The secondary structure is the local helices and each chain. The tertiary structure is the folding of the alpha chain. And the quaternary structure is the triple helix formed by the 2 COL1 chains and 1 COL2. And for the R836C mutation, the impacts of all these because even one wrong amino acid disrupts the folding and weakens collagen's integrity. Now we're going to talk about the functional impact of cells and organisms. In a healthy person, the collagen fibers are strong, flexible, and long-lasting, but with the R836C mutation, the fibers are brittle and disorganized. In Capi's disease, this causes thickening of bones, especially jaw, ribs, and long bones, pain and swelling, fever, irritability in infants, and in some cases, long-term skeletal deformities. Now let's talk about how I physically built this model. I used white paper strips for the collagen chains. I marked the R836 or C836 sites using colored paper dots. So these are what they look like. And for the mutated model, I loosened the twist and added paper clips between the chains to represent an abnormal disulfide bond. So these are the paper clips that I added. And the normal model is neat and stable, so make it super tight. And then the mutated model is more tangled and unstable and uneven, so it looks more like this. Just like a misfolded collagen in Cappy disease. So for the limitations in my model, every model has limitations, and here are 3 for mine. 1 is the real proteins fold at the molecular level and not just paper and clips. The model is static, but in real life the proteins are dynamic and constantly interact with each other's models. And 3rd, each chemical interaction like ionic bonding, calcium binding, and enzymatic processing aren't represented in the simplified build. But still, this model is a powerful way to visualize the structural consequences of single point mutations. Thank you.